Hello, my name is Stephanie Rossi, and this is a presentation of my work titled Metabolic Regulation of Gamma-aminobutyric acid during Heat-Induced Leaf Senescence in Creeping Bentgrass. Cool season turf grasses are economically important in the United States and are widely used throughout the country. The optimal temperatures for cool season grass growth and development range from 60 degrees Fahrenheit to 75 degrees Fahrenheit, and they are sensitive to temperatures above this range. In summer months, cool season turf experiences heat stress, which induces and accelerates leaf senescence, a process that is hallmarked by yellowing and thinning of the turf canopy and results in loss of chlorophyll. Nitrogen is an important compound that plants use to synthesize amino acids, but nitrogen metabolism is limited by heat stress, which affects amino acid metabolism. Gamma-aminobutyric acid, or GABA, is a nitrogen-rich amino acid that has a role in carbon-to-nitrogen balance and accumulates under various abiotic stresses. Additionally, it has been shown to increase chlorophyll content and protect plants against oxidative stress, which enhances stress tolerance. Despite there being documentation of the positive effects that GABA imparts on plants exposed to abiotic stresses, the manners in which GABA specifically affects chlorophyll or amino acid metabolism is not well understood. To determine how GABA may alleviate heat-induced leaf senescence, our objectives were to investigate the effects of exogenously applied GABA on leaf senescence and to determine the metabolic factors regulated by GABA that contribute to the mitigation of heat-induced leaf senescence in creeping bentgrass. Sod plugs of mature creeping bentgrass of the cultivar Pencross were established in pots that had a 40 centimeter depth and a 10 centimeter diameter containing fritted clay. Plants were established for 28 days in a greenhouse, after which they were moved to environmental growth chambers controlled at 22 degrees Celsius in the daytime and 18 degrees Celsius at night, 60% relative humidity, and a 14-hour photo period of 750 micromole meters per second squared photosynthetically active radiation. The study was arranged in a split plot design with temperature treatments as the main plots and individual chemical treatments as subplots. Each treatment, controller GABA, had 6 replicates that were arranged randomly within each growth chamber, and plants were relocated across 4 different growth chambers every 3 days to minimize variations in chamber environments. To impose the heat stress treatment, plants were exposed to 35 degrees Celsius in the daytime and 30 degrees Celsius at night for 35 days. The non-stress control plants were grown under optimal temperature conditions at 22 degrees Celsius in the day and 18 degrees Celsius at night. Plants were foliar sprayed to canopy saturation with 50 milliliters of water for the untreated control or 0.5 millimolar of GABA prior to exposure to heat stress and then weekly during heat stress. Turf quality was visually evaluated on a scale of 1 to 9 according to canopy density, texture, uniformity, and greenness. A rating of 9 was assigned to turf that was uniform and green, while 1 indicated wilted, brown, and desiccated turf. 6 was the minimal value at which turf was rated acceptable. To determine leaf chlorophyll content, fresh leaves were submerged in dimethyl sulfoxide and incubated under dark conditions for 72 hours. Absorbance of the chlorophyll extract was measured at wavelengths of 663 and 645 nanometers using a spectrophotometer, and the dry leaf tissue was weighed on a mass balance so that chlorophyll content could be calculated. Activity of the chlorophyll-degrading enzyme chlorophyllase was measured. To do this, chlorophyll was extracted and purified from the leaves of non-stressed creeping bentgrass, and it was mixed with an extraction buffer derived from each sample plant. The mixture was used to simulate the conversion of chlorophyll to chlorophyllide, which is catalyzed by chlorophyllase. This is a rate-limiting step during chlorophyll degradation, as it causes major changes to the structure of the chlorophyll molecule when the phytol chain is removed to yield chlorophyllide. To examine whether the effects of GABA on leaf senescence were related to alterations in amino acid metabolism, The endogenous content of amino acids was analyzed at 35 days of heat stress using hydrophilic interaction, ultra-high performance liquid chromatography with tandem mass spectrometry. The effects of chemical treatments and temperature on all measurements were determined by the analysis of variance test using SAS version 9.2. To separate differences between treatment means, Fisher's protected least significant difference test was performed at the 0.05 probability level. Turf quality and chlorophyll content declined overall in plants exposed to prolonged heat stress, but to a lesser extent in plants treated with GABA. Under heat stress, GABA-treated plants maintained significantly higher turf quality at 28 and 35 days of heat stress, while chlorophyll content was significantly higher in GABA-treated plants at 14, 21, and 35 days of heat stress. Conversely, the activity of the chlorophyll-degrading enzyme chlorophyllase increased through the duration of heat stress, but its activity was significantly lower in plants treated with GABA from 7 days through 35 days of heat stress when compared to untreated controls. To further understand the metabolic effects of GABA on heat-induced leaf senescence, we examined which amino acids may have been responsive to GABA treatment under heat stress and whether there are any metabolic pathways associated with amino acids that may have been regulated by GABA during heat-induced leaf senescence. The endogenous content of glutamic acid, threonine, and GABA was significantly higher in GABA-treated plants as compared to untreated controls under heat stress, whereas none of the other amino acids examined were significantly altered under heat stress. Glutamic acid, threonine, and GABA are all active in the GABA shunt pathway, a process that bypasses 2 steps in the tricarboxylic acid, or TCA cycle, and expedites the conversion of alpha-ketoglutarate to succinate. In the GABA shunt, glutamate is converted into GABA in a reaction catalyzed by glutamate decarboxylase, or GAD. GABA is transaminated into succinic semialdehyde, which then becomes succinate. By systematically channeling glutamate into the TCA cycle, the GABA shunt expedites energy production. In conclusion, GABA suppresses heat-induced leaf senescence through the regulation of both chlorophyll and amino acid metabolism. More specifically, applying GABA to creeping bentgrass under heat stress elevated chlorophyll content by decreasing the activity of chlorophyllase, a key chlorophyll degradation enzyme. Additionally, it increased the levels of amino acids having metabolic functions in the GABA shunt pathway, suggesting that it improved energy production in the TCA cycle. In the future, it may be possible to use the amino acids specifically responsive to GABA as biomarkers for the selection of heat-tolerant germplasm in cool season turfgrass species, and these amino acids may also be incorporated into biofertilizers used for turf management in order to improve heat tolerance in warm climates.